Hello, and welcome to Science in Seconds, where we break down complex scientific concepts into bite-sized pieces. Today, we're going to talk about genetics, the study of genes and how they're passed down from one generation to the next. We'll also delve into gene editing, a revolutionary technology that has the potential to change the way we think about treating genetic disorders. To understand genetics, we need to first understand DNA. DNA is short for deoxyribonucleic acid, and it's the genetic material that makes up all living things. DNA is made up of four building blocks called nucleotides: adenine, guanine, cytosine, and thymine. These nucleotides pair up in a specific way, with adenine always pairing with thymine and guanine always pairing with cytosine. This pairing forms the famous double helix structure of DNA. Genes are specific segments of DNA that contain the instructions for making proteins, which are the building blocks of life. Genes determine everything from our physical characteristics, like eye color and height, to our susceptibility to certain diseases. Genes are passed down from our parents, with half of our genetic material coming from our mother and half from our father. Gene editing is a technology that allows scientists to modify DNA in a precise and targeted way. This technology has the potential to revolutionize the treatment of genetic disorders, such as sickle cell anemia and cystic fibrosis. One of the most promising methods of gene editing is CRISPR Cas9. CRISPR is short for clustered regularly interspaced short palindromic repeats, which are unique sequences of DNA found in bacteria. Cas9 is a protein that can cut DNA at specific locations, guided by RNA molecules that match the targeted DNA sequence. With CRISPR Cas9, scientists can essentially cut and paste DNA, replacing a faulty or mutated gene with a healthy one. This technology has been successfully tested in animals, and human trials are currently underway. While gene editing has the potential to cure genetic disorders, there are also ethical considerations to take into account. One concern is the potential for gene editing to be used for non-medical purposes, such as creating designer babies with specific physical or intellectual traits. There are also concerns about the long-term effects of gene editing, both for the individual being treated and for future generations. Changes made to DNA can be passed down to offspring, potentially affecting the genetic makeup of entire populations. The study of genetics is rapidly advancing, and new technologies and discoveries are constantly being made. In addition to gene editing, other promising areas of research include epigenetics, which looks at how environmental factors can affect gene expression, and personalized medicine, which tailors treatments to an individual's unique genetic makeup. There you have it, a brief overview of genetics and gene editing. While there are still many unknowns and ethical considerations to take into account, it's clear that genetics is a rapidly advancing field with the potential to change the way we think about medicine in the human body. Thanks for watching, and be sure to subscribe to Science in Seconds for more videos on the latest scientific discoveries.